All right, we are on lesson 2 of chapter 13. So once again, clicking the wrong button. In this lesson, we are looking at the actual process of protein synthesis, the process of how we are really making our proteins from the mRNA that we discussed in the previous lesson. So we're going to start by talking about just the genetic code in general. So the genetic code, the purpose of that code is to be able to make proteins. And remember, proteins are also called polypeptides. So if you see that in certain sources, they mean the same thing. Okay. A protein is a specific sequence of amino acids. So we talked about the tRNA and how it brings in the amino acids. And so it's that order of amino acids that's really important. That's what makes your protein. If you have the wrong order of amino acids, your protein probably won't work right. And that causes lots of diseases and disorders. Okay. The sequence of amino acids is coded in your DNA. So your DNA contains the code. If your DNA is messed up, that's how you have a genetic condition that causes you to have proteins that are not correct. And we're going to talk about some of those in chapter 14. The code from your DNA, right? So your DNA actually gets turned into mRNA, right? You make that mRNA copy and change the DNA into the mRNA. It makes a copy. And the code is read 3 bases at a time. We call that a codon. Okay, so this is a picture from your book where you have, this is representing the mRNA, and you have 3 bases together, they make a codon. So each codon is going to code for 1 amino acid. All right, and the way that it does that is you have the codon of your mRNA, and it is complementary to the anticodon on the tRNA. So we looked at that kind of loopy, clovery structure to the tRNA, right? And that bottom loop has the anticodon. Again, it's complementary to the codon of the mRNA. So if your mRNA says UCG, U pairs with A, C pairs with G, G pairs with C. So they're complementary. And then this anticodon matches a particular amino acid. So the other end of the tRNA is kind of specifically made to fit a particular amino acid. And so it can bring in the correct amino acids. With 4 bases, and you're putting them together in sets of 3, and they can repeat, right? You can have AAA. Or AAG or anything like that. You have actually 64 possible combinations of bases. Okay, so from the 4 bases being put together in any kind of order of 3, you get 64 possible combinations. But there's only 20 amino acids that get used in, at least in eukaryotes. Okay, most living things only use these same 20 amino acids, which means that we have what's called redundancy. So if you don't know the word redundant, it means like saying something the same way in a sentence, right? So it's redundant. Okay, you've said the same thing, or maybe you say the same thing 2 different ways in the same sentence. And so, but you've said the same thing. Okay, so our code is redundant because some of our amino acids can be coded for by more than 1 codon. More than 1 codon can bring the same amino acid. And that's going to be a good thing for us in a little bit. We'll see that when we talk about mutations and some of the errors. So this is called a codon wheel. There's also a chart. I think we look at the chart in a second. The codon wheel, you have a picture in your book, but it's not color-coded like this. So you start from the inside and go to the outside. So if your codon was ACA. You start here at the middle, you go A, C, A. And so ACA brings THR. So these are the abbreviations. I just happen to know this one is threonine because I've studied this a lot. And so some charts will have the whole name, some charts will just have their abbreviations. Use whatever you have, okay? But you can see that ACA, ACG, ACU, and ACC all bring the same amino acid. Okay, so that means that if we have a mutation in this third spot, it doesn't matter. We're still going to get the same amino acid, and that's going to be good because then we can still make our same protein. Awesome. Okay. A couple other things to point out. We do have a start codon. So the first codon for every every protein chain always starts with methionine, MET. So the first codon is always AUG. And then there are 3 stop codons. You can see them here. So UGA, UAA, and UAG can all code for stop. And stop actually doesn't bring another amino acid. It brings in kind of a blank. And so there's nothing there. And so it stops attaching things. And your protein is made, or the chain is made. Okay, so there's a couple of things there to point out. The codon chart works like this. So you start over here with with the first position, and then the second, and then the third. You can also just kind of look within and find the one you're looking for. It's almost sometimes easier. So if it's GCA, G, second position is C, looking for GCA right here, and this one is ALA or alanine. All right, so the start and stop codon. So each codon is almost like a word. Right, so we have 3-letter words that we put together into a sentence. So methionine is always the start. And then you have 3 stop codons that are the end. Okay, so AUG always starts. And then there are 3 different stop codons that you could have to terminate your protein creation. All right, so the process of reading the code, reading the codons and creating the protein is called translation. Okay, decoding that mRNA message and actually making the protein. So we start with our RNA codons. That brings in the specific amino acids, which get bonded together to make the protein. And that's translation. Translation is going from the RNA codons to the protein. I have lots of images for this. Okay, so here we have our ribosome in the dark blue. And then you have the mRNA strand. And so we're reading that 3 letters at a time, starting over here. And then adding the particular amino acids in the right order, bonding them together. And then once you get to the stop codon, it would be released. Okay, so showing the whole process of transcription, right? Going from DNA, making our RNA that gets transported to the cytoplasm, finds our ribosome, and we have translation occurring where we're adding our tRNAs. tRNAs are coming and bringing the correct amino acids, adding those amino acids to the polypeptide chain, and then they go away to go find more amino acids. Okay, and one more. Messenger RNA, the mRNA, with its codons that are complementary to the tRNA anticodons. tRNA brings in a particular amino acid and adds it to the chain, and then goes away to go find another amino acid. Okay, and again, showing the same thing. We do DNA to mRNA, and that happens in the nucleus. Good job. And then the mRNA leaves the nucleus, goes to the cytoplasm, finds a ribosome, either a free ribosome or one that's attached to the ER. And the mRNA binds to the ribosome and the tRNAs bring in their particular amino acids, adding them to the chain. And then that chain is going to wind up and curl up and do all the different structural things that we talked about last semester. Okay. All right, this is what we call the central dogma of biology. So a dogma is like a central idea, okay, the main idea of what's happening. So this is the central dogma of how information moves or how information flows in biology, that we go from our DNA to the mRNA to protein. So we go genes to messenger to protein. Okay, so that order is very specific. So we do DNA to mRNA. So this arrow is representing which process? Transcription. Okay, and then mRNA to protein is representing translation. Okay. So the way that our genes are actually put into action is what we call gene expression. So some genes get expressed more often than others. And some of that depends on your own DNA and what you need at the time and your environment, and all sorts of factors go into determining what we actually express in terms of our genes. Okay, so gene expression is just how your DNA and your RNA and all of your proteins are involved in taking your genetic information and making that into you. Okay, so that's gene expression. We consider our genetic code to be universal because pretty much all organisms use the same code and they use it in the same way, reading those 3 bases at a time. It gets read in the same direction. So there are just very, I lost my train of thought there. I don't know. But it's used by almost everything. There are a handful of viruses that are called retroviruses, and they contain RNA instead of DNA. But most organisms contain DNA. The DNA gets transcribed into RNA, and the RNA gets translated into a protein by reading the 3 codon or 3 bases for the codon, and they're read in the same direction. Okay, so that's what we mean by universal genetic code. All organisms are using the same code and in the same way, which is kind of important. Okay, so that is the end of lesson 2. Again, send me questions if you have any. Let me know how things are going. You guys might be a little bit behind. I don't know. You have 2 labs actually to do with this lesson. No, you have 3. Cool. Yeah, so you have 3 labs that you're going to do with this lesson. So the first one, you're going to do cut and paste, and I'm going to do separate videos to show you kind of how to do those. So one is a cut and paste lab, and then one is more just paper-based, just an analogy of how we can take the DNA code and turn it into a message. And then we have another one where you're actually going to model the process. And that's one that I kind of created and put together myself. So hopefully I've been using it for a few years. So it's pretty good. It's pretty good. So those are 3 labs, or the 3 activities that we're going to do with this lesson. And then we'll move on to lesson 3, which is mutations.